waterproofing. The epicuticle, it acts as an waterproofing, and it protects the cuticle from chemically harsh treatments like chemicals. And it also acts as a lubricant, like a smooth movement for the prevention of frictions from affecting the insect's body surface. And this is the cement layer, where you can see the upper one, the epicuticle, and this is the wax layer. Okay. So this is basically how it looks. The cuticle, this is the epicuticle where you have the cement and the wax layer. And this is the exocuticle. Endocuticle is like the outerwards. It is together exocuticle and endocuticle together called as procuticle. And the down you will have the epidermis with basal lamina, gland cells, everything will be present. So together this epicuticle, exocuticle, endocuticle, this all the three layers together are called cuticle. So here we are talking about procuticle. We have seen the procuticle, right? From here to here, it's a procuticle. So what we'll see, procuticle is beneath the epicuticle. So epicuticle is the outermost layer we have seen in the cuticle. So procuticle will be just below the epicuticle. It is the thickest layer, and it's responsible for providing structural integrity to the exoskeleton. It is made up of, again, two layers, the exocuticle and endocuticle. Here the procuticle is made up of exocuticle and endocuticle here. The exocuticle is largely composed of a chitin, which is a long-chain polysaccharide, proteins, and sometimes minerals such as calcium salts. The protein-chitin matrix is often cross-linked to provide rigidity and strength. And functions of the exocuticle is it gives strength, and it is also used in molting. Molting is a process where the exoskeleton, the old exoskeleton will be replaced with the new exoskeleton. This is the process that we often see in most of the arthropods. Coming to the endocuticle composition is it contains chitins, proteins, and substances, whereas it is less rigid than the exocuticle and is often more flexible. So if it's not rigid, then it has a tendency of flexibility, right? So flexibility, what does this flexibility do? How does it help the cuticle layer in insects? So this layer allows the cuticle to bend and stretch, especially in areas where movement is required, like joints, right? So in that places, it has to be flexible, because if it is rigid, then it cannot bend or stretch. So rigid in the sense, it's like a concrete, like a very strong one. No movement will be there. So you cannot move it. It is like a rigid one. Flexible in the sense, you can easily move it, right? Look. So this layer, the endocuticle, is flexible, allowing the movement of the cuticle. And the other function of it is also growth. The endocuticle is involved in insects' growth, because if stretching is there, then only the insect size will also grow. And molting process. So molting process is like, if in order for an insect or any arthropod to grow in size, the molting process has to happen, where the shedding of the old exoskeleton takes place and formation of the new exoskeleton will happen, providing new material for the exoskeleton. So the molting is the process that most of the insects we see. And for example, in a butterfly, if we see, so initially it will be in a different stage, but later on it turns out to be a butterfly using this molting stage. So summary of layers and functions here in detail we'll discuss, like epicuticle is composed of wax and cement layers responsible for waterproofing, chemical protection, and reducing the friction. Exocuticle is also composed of chitin and proteins, but it provides rigidity and strength to the cuticle, whereas endocuticle, it provides flexibility and aids in growth during molting. Together, these layers ensure that the insect's survival by providing protection from physical damage, dehydration, because the water loss will be prevented, and harmful environmental factors. So it provides protection from harmful environmental factors while allowing for growth and movement. Coming to the role of chitin. So we have seen that all the layers of the cuticle, so if you see here, all the layers of the cuticle, they are initially made up of chitin. Chitin is a long polysaccharide. So this is made up of a chitin. So what is this chitin and what is its role in the insect cuticle? We'll discuss here. So chitin is a key structural polysaccharide which is found in the cuticles of insects, which provides mechanical strength and rigidity. It is the major component of the exoskeleton, contributing significantly to the cuticle's overall dry weight. Coming to the constitution of the dry weight in insect cuticle, so chitin composition. In the insect cuticle, chitin constitutes up to 30 to 50% of the dry weight of the cuticle. So you can see the majority of the cuticle is made up of the chitin. The exact percentage can vary based on the insect species. So it varies from species to species. The percentage of chitin will vary from species to species in the cuticle. For example, more chitin may be present in areas of the exoskeleton that need additional strength in different species we are talking here. Other compounds, in addition to chitin, that are present in the cuticle are proteins, which can make up to 50% of the dry weight. Lipids, mainly they are present in the epicuticle, and minerals such as calcium salts, particularly in certain insect groups like crustaceans or certain hard-shelled beetles. So coming to the chitin structure. So we are talking about chitin and its importance and its composition and how much of composition is there in the cuticle. So we will see what is the chitin and what is its structure is like. Okay. So chitin is a polymer. It's a polymer made up of repeating units of N-acetyl glucosamine, a derivative of glucose. So here chitin is a polymer. But what is a polymer? If many monomers combine, then we form a polymer. That will form a polymer, right? So here, N-acetyl glucosamine. So glucosamine is a saccharide, right? It's a monosaccharide. So uh, the amine group, when attached, when it undergoes acetylation, then the N-acetyl glucosamine will be formed. This is like a sugar, ribose sugar will be present here. And N-acetyl glucosamine is a derivative of glucose. So if one N-acetyl glucosamine will get linked to the another N-acetyl glucosamine, and together like too many, they form and forms a polymer of chitin. So that linkage is called, that linkage and that strength together making up of more monomers build up to form a polymer. Okay, so the basic unit of chitin is a disaccharide, which consists of two molecules of chitin. So chitin is basically a disaccharide consisting of two molecules. So one acetyl glucosamine is a monosaccharide. If two are present, then it is disaccharide. If more than two are present, then it is polysaccharide. No. So in a similar way, when two molecules of N-acetyl glucosamine, they combine to form a disaccharide, which is the basic unit of the chitin. And in order to join these two molecules, we need some kind of bond, right? So there should be some bond to bond these two molecules. So one molecule, when it has to bind with the other molecule, there should be some kind of interaction or there should be formation of some bond. So like a covalent bond, that function will be done here by the linkage of the beta-1,4 glycosidic bonds. Beta-1,4 glycosidic bond. So it is like a covalent bond that connects sugar molecules, that is like glucosamine molecules, a monosaccharides, to another molecule, which can also be another sugar or a different type of molecule. But here in case of chitin, it is the same molecule. So beta-1,4 glycosidic bond. So here we have a beta-1,4 glycosidic bond. So this is very important. So because chitin structure is also similar, mostly similar to that of the cellulose structure that we basically see in the plants, where the cellulose is also made up of beta-1,4 glycosidic bond. The difference is that here we have N-acetyl amino group in the position where there will be hydroxyl group in the cellulose. Okay, so this beta-1,4 glycosidic bond formed between an anomeric hydroxyl group of one sugar that is in the beta configuration and hydroxyl group of the fourth carbon atom. So here you'll have a structure of the chitin. So basically this is it. So the glycosidic bonds together binds to glucosamine anestel glucosamine molecules. So this is the anestel group that is this is the anestel group here, and this is the anestel group here. So these are two different molecules which are bound by beta 1,4 glycosidic bond. So this is the first carbon position here, and there is a second carbon, third, fourth, fifth, and sixth. No, so here this first the one from one sugar molecule, the one first position of the carbon, and when it comes to in this way, this will be the fourth one. So 1 and 4 will together link up to form 1,4 beta glycosidic bond here. So the fourth carbon of the another sugar molecule creates a disaccharide or large polymer with repeating units, like these are all the repeating units held together by this specific glycosidic linkage. The same thing we can also get to see in cellulose, we talked earlier, right? Yeah. Polymeric structures. The polymer is a linear consisting chains of N-acetyl glucose amine units that are highly organized into a crystalline structure. So this crystalline structure contributes to the strength and rigidity of the exoskeleton. So together, all these N-acetyl glucose molecules together, they form a polymer. Polymer means a big linear molecule, polymeric structure, which is very much rigid, which gives the strength and rigidity to the exoskeleton, making the outer cuticle very strong and rigid. Okay. The chitin unit cell and crystallinity. So unit cell of chitin, the unit cell of a chitin refers to the smallest repeating structural units of the chitin molecule, where two N-acetyl glucose. So we have discussed this. We have I've explained this. The chitin forms a crystalline region such as chitin fibrils. They are amorphous, means they will not have a defined structure. But later on, upon the addition of more number of molecules, they tend to become more rigid. And polymorphism of the chitin. Chitin exists in several forms like alpha, beta, and gamma. Whereas alpha chitin being the most common form in the insect cuticle, you tend to see alpha chitin the most. The crystal structure of alpha chitin is composed of parallel sheets arranged in a sheet-like configuration, which gives it a specific structure like rigidity. So due to the arrangement of these alpha sheets in the chitin structure, the rigidity and the strength will come to the cuticle. Uh, so this everything I have explained here, the structure also we have seen here. Coming to the synthesis of chitin, yeah, fine. So the chitin has very important and prominent role in it. It gives us a rigidity and structure of it. So how does this chitin get synthesized? So that we are going to see here. So chitin synthesis is a highly regulated biological process that occurs in the epidermal cells of the insects. So where does this chitin synthesis happen? In the epidermal cells of the insects, particularly during the formation of the cuticle. And this involves several key steps, and there are enzymes that are involved in synthesis of this chitin, the chitin synthase enzyme. So what does this enzyme do? The enzyme chitin synthesis is responsible for catalyzing the polymorphism of N-acetyl glucosamine. So it will help the joining of the simpler units, the monomers of N-acetyl glucosamine into a long polymer linear chain. So this enzyme works by linking the N-acetyl glucosamine molecules through beta-1,4 glycosidic bonds. So the function of chitin synthase enzyme is to link up the N-acetyl glucosamine molecules by using a beta-1,4 glycosidic bond. So that is how the synthesis happens. And the precursors for this are the primary precursor of chitin synthesis is uridine diphosphate N-acetyl glucosamine. So UDP, uridine is a nucleotide and which has two phosphates. Are you hearing? I think I'm visible and audible, so I'm continuing the class. Here I just got struck in the video. So chitin synthase is the enzyme that works by linking N-acetyl glucosamine by beta 1,4-glycosidic bonds. We have read this, and the precursor is UDP and acetyl glucosamine. It's a nucleotide sugar is synthesized from glucose and serves as the building block of chitin. So coming to chitin synthesis pathway, so synthesis of UDP. So initial the precursor molecule that we have talked here is the UDP and acetyl glucosamine. So glucose is converted into UDP glucose, which is then converted to UDP and acetyl glucosamine. Then polymerization by chitin synthase happens. The enzyme chitin synthase facilitates the addition of UDP and acetyl glucosamine to the growing chitin chain. And once synthesized, chitin chains are transferred to the extracellular space where they assemble into large fibrils that integrate into cuticle matrix. And this is the chitin synthesis pathway here. So first glucose is converted into glucose 6-phosphate. So the primarily two steps or three steps are moreover similar to the glycolysis pathway, where you have in trihalose the 6-phosphate, which gets converted to glucose 6-phosphate, and by the enzyme glucose 6-phosphate isomerase that gets converted to fructose 6-phosphate. But whereas in glycolysis it gets converted to fructose 1,6-bisphosphate, but here with the help of glutamine fructose 6-phosphate enzyme, the phosphate groups get attached and the glucosamine 6-phosphate is formed, and then glucosamine 6-phosphate acetyl transferase. The acetyl group from the coenzyme A will be transferred. Acetyl CoA will be transferred to glucosamine 6-phosphate, making it N-acetyl glucosamine 6-phosphate. And phosphoacetyl glucosamine mutase converts it to N-acetyl glucosamine 1-phosphate. Upon UDP N-acetyl glucosamine pyrophosphorylase enzyme, it will make N-acetyl glucosamine 1-phosphate. It converts it to UDP N-acetyl glucosamine. So UDP N-acetyl glucosamine, as we have talked, it is the precursor of the chitin. So this is how the chitin will be made. From the UDP N-acetyl glucosamine, the chitin synthesis will takes place. So this is the same information that I have given here for the purpose of the notes. Coming to the function of chitin in the insect cuticle, structural integrity. What does chitin help, or what is the function of the chitin in the insect cuticles? Yes, insects have a cuticle, and cuticle is majorly made up of a chitin. So what is the use of chitin? So it gives the structural integrity. Chitin provides the insect cuticle with its structural rigidity and strength, enabling it to support the body and protect against physical damage. Okay. Coming to the protection, the chitinous exoskeleton protects the insect from environmental threats, including predators, pathogens, and mechanical damage. Support for growth and movement. Chitin also enables the exoskeleton to maintain its shape while allowing for movement at joints where the cuticle is thinner and more flexible. Some chitin, so